Hi there, welcome back to my channel Xgene proteinX. In this tutorial video, I'll discuss about the elongation process in eukaryotes. So in my previous video, I already discussed about the initiation process in eukaryotes. So before going to the main discussion, I'd like to have a quick recap that how initiation occurs. So first, the DNA helicase enzyme that recruited in the pre-initiation phase, and the DNA helicase, it separates this DNA strand from each other. Actually, the main function of the DNA helicase is to unwind the DNA strand. Afterwards, these bubbles forms. So these bubbles also known as the origin of the replication, because the replication start inside these bubbles in the both direction. So thereafter, the RNA primer it recruited by the primase enzyme, and RNA primer it bind in the 3' region in the both strand. So always remember that RNA primer always it bind in the 3' region, and then the DNA polymerase alpha it recruited by the CDC45 protein. So upon binding the DNA polymerase alpha, the start the initiation process. So then the DNA polymerase alpha it starts synthesizing the new strand around 15 to 20 nucleotide long stretch of the DNA strand towards the 5' region, and then the elongation start from here. So next move on to the elongation part, but before that I'd like to make one quick discussion that how this DNA helicase works. So you see here this picture, this the strand it's separated by the DNA helicase. So DNA helicase actually it bind here, it works here towards this direction. So these two strands separated then, and the replication fork it work or it keep going on in this direction. So this is known as the replication fork, and then in this strand, both of the single strand, then the primer bind in the 3' region. So here green color, and here also green color. So then the replication start in this direction for this strand, and another strand in this direction, opposite direction. Alright, so this is the leading strand, and that is the opposite one is the lagging strand. So I'll discuss later about this leading strand and the lagging strand. So let's move back to the main discussion. So we are here that elongation process it start. So next is the Pcn protein that recruited. So Pcn proteins already is recruited in the pre-initiation steps, but Pcn proteins, the main function of the Pcn proteins, it help for the detachment of the DNA polymerase alpha, as well as it recruits the DNA polymerase epsilon and DNA polymerase delta. So Pcn proteins it has two different job actually. So upon the recruitment of the DNA polymerase epsilon and DNA polymerase delta, it keeps adding the nucleotide sequence from the DNTPase in the both strand. So mainly the DNA polymerase epsilon, it is dedicatedly replicate for the leading strand. So this one is the leading strand, and for the DNA polymerase delta, that is for the lagging strand replication. So as you see in this figure, in the leading strand, the RNA primer is only once it recruited, it bind here only once, and then it continuously the DNA strand synthesizing. But for the DNA polymerase delta, there is some problem, because there are multiple RNA primer it binds, and there are short segments of the DNA strand it formed. Alright, so then I would like to mention here that how this kind of DNA nucleotide it adds, and then how this kind of bases are extended. So let's move on to have a quick discussion that how this DNTP works. So as you see in this figure, the DNTP is known as the deoxyribonucleotide triphosphate. So it contains triphosphate because it contains 3 different phosphate groups, and there is one ribose sugar, and here is the bases are attached in this position. So different groups and different bases is attached. So for A, T, G, C is different DNTPs. So actually the DNTPs is the mixture of these 4 different bases. Alright, so here in this figure, as you see, these 2 bases already it's joined together. So this is the 5' region and this is the 3' region. So DNA polymerase, it works in this direction, towards this direction, 5' to 3'. So here you see this the one of the strand. Here you see the cytosine, this base it attached here, the phosphate group it attached with this O hydroxyl group, and then phosphodiesterase, this linkage it formed, and then one of the 2 different inorganic phosphate ion separated. So this is the reaction that how this strand extended, and after that eventually the strand, you see, from with this A and T, this both base pair, the another base pair is joined together. So these bases are extended. So this is how DNA polymerase work, by adding nucleotide sequence. So let's move back to our main discussion. So we are here. Sorry, another important things I want to mention here, that the enzyme ribonucleotide reductase is the key enzyme that supply the DNTPase. So the main enzyme, the role of this enzyme is to synthesizing DNTPase that is required for the adding nucleotide in the DNA replication. So okay, so we are here that the DNA polymerase delta and DNA polymerase epsilon, it starts synthesizing the strand by adding nucleotide one by one, right? So next step is the dissociation of the primers. So as you see in this figure, there are a lot of primers binding here and here also one primer. So this is actually the DNA strand, right? So the RNA primer should be out of the DNA strand. It must be, right? Otherwise it will be the DNA RNA complex, DNA RNA duplex formed. So that is why the RNase H and FEN1, these both enzyme, they remove the RNA primer. So what they do, the RNase H, this enzyme, first it detect the RNA DNA duplex or RNA DNA hybrid, and then it degrades the RNA and along with some ribonucleotide. So after that, FEN1, this enzyme, it removes the rest of the ribonucleotide from the DNA strand. Then it would completely free of ribonucleotide from the DNA strand. So then the completely out of the ribonucleotide from the DNA strand, it occurs by these 2 enzymes. So mainly the 2 enzymes, they remove the RNA primer from the DNA strand. Alright, and then as you see in this figure, there are some gaps formed, and also there are some short segments it formed for the lagging strand, not in the leading strand. And these short segments are known as the Okazaki fragment. Alright, So let's have a look. So here I would like to mention that how it forms. So actually this is the one strand that's separated, and then this is the double strand, and here the DNA helicase, it works in this direction. So this is the DNA helicase, so it works in this direction. So then in the 3' region, the primer it bind here, and then DNA polymerase it start synthesizing the new strand towards the 5' direction. So for the leading strand, this is the leading strand. So for the leading strand, there is continuous synthesizing the new strand, because this is the direction, the same direction of the helicase works, that in the same direction. But for the lagging strand, that is a problem actually, because it works in the opposite direction, this direction. So what happens here? Upon unwinding this DNA strand, this one stretch of the DNA is come, and then primer binds somewhere here in the 3' region. So after that, the DNA polymerase come and it synthesizes this new strand, right? So next step, it will be clear if I draw it completely. So here what happens? So the DNA helicase, it works further and it unwind the another stretch of the two DNA strand, right? Then you see here, this is already this stretch of the DNA strand, it already formed previous step. So here is the DNA strand. So upon unwinding this DNA strand, then another primer recruit here, another primer it bind here, and then the DNA polymerase comes and bind here, and then it starts synthesizing the new strand in this direction, because this is the 3' region and this is the 5' region. So it only synthesizes only this small segment, because in the previously it already synthesized this part, right? So this part already synthesized this part, so only this part is synthesized newly. So afterwards again the DNA ligase works further and it so then it already here one strand that is previously formed, here is another strand, then upon stretching out the another strand, then another RNA primer it recruit here, and then DNA polymerase it binds here, and then it works in this direction, start synthesizing. So you see here how this kind of small fragment it created. Alright, so these small fragments are known as the Okazaki fragment that I mentioned here. Like, okay, let's move on to the next part. So we are here that RNase and FEN1, that both enzyme that removes the primer, RNA primer. So upon removing the RNA primer, these kind of gaps were created, right? These gaps are created. So here the small gaps, and here only one gaps are created. So next step is again the polymerase delta it start working to synthesize the another small segment of the DNA strand to fill up that gap that created by the removing of the primer. So here the one segment here, and here the small part, that gap created, that is filled by the the DNA polymerase delta. Alright, then this kind of new strand, it comes and it fill the gap. So next process, but still there are some problem, because there are some gap in between the newly synthesized strand, because there are some nick, the phosphorylase linkage is not formed in between. So that is why ligase enzyme it comes, and then ligase enzyme it joined the nick, and then it formed the phosphorylase linkage, and then eventually the new strand, complete new strand it formed in both sides. Right? So this is how this elongation works. But I also would like to mention, and also like what I mentioned in this elongation step, I just precisely would like to discuss here. So this is everything about the elongation step. Just have a quick discussion here. So what happens? The helicase enzyme it comes here, and then helicase enzyme it works in this direction. So then this strands it separated from each other, and then the primase enzyme it bind in both side, and then in the RNA primer it recruited by the primase enzyme. Then RNA primer it bind here, and then the DNA polymerase it starts synthesizing new strand in this direction. Alright, so then there are some small segment, and these small segment are joined by the DNA ligase enzyme. Also, it also in the same things also happen in this side, but in the opposite direction. So this is in this side, and this is in this side. So alright. So here another thing is that here is one single strand binding proteins also here, and their main job is the single stranded binding proteins is to protect the DNA strand from the cleavage. And also another important key enzyme that works here, the key role of this topoisomerase, this topoisomerase it relaxes the supercoils formed of the DNA by cutting one or two strands, and help DNA polymerase to synthesize new strands smoothly. So this is the main function of the topoisomerase. So this is all about the elongation process. So I hope this video will be helpful, and if you find this video help you to understand the elongation process properly, so kindly hit the like button, share it, and subscribe my channel. Thanks.